Hello there, my name is Pelumi. In this audio, I will be presenting my summer research. This summer, I researched the role of septin 7's phosphorylation by tau K2 in neuridogenesis and how this mechanism could potentially contribute to the prevention of neurodevelopmental disorders such as autism spectrum disorders and schizophrenia. Neuridogenesis is the formation of neurites, the precursors of axons and dendrites in neurons, and is crucial for proper signal transmission in the nervous system. Previous studies have shown that defects in the cytoskeletal mechanisms of neuridogenesis underlie many neurodevelopmental disorders. There are 13 known mammalian septins categorized into subfamilies based on sequence similarity. The only member of its subfamily, septin 7, whose role has been implicated in the regulation of neuridogenesis as well as cell body extension and retraction, needs to be phosphorylated and properly localized for it to perform its function. This phosphorylation of septin 7 is therefore performed by protein kinase known as tau K2. Per previous research, tau K2 uses its K67 domain to phosphorylate septin 7. Figure 1 explains the stages of neuronal development as has been characterized in vitro, that is, the cells were cultured in a dish. Neurons generally start around and without neurites known as the IV0 and begin to develop protrusions which become neurites between the IV0 and 1. By DIV3, neurites develop growth cones which drive neurite extension and begin to undergo axon-dendrite polarization. So to answer the research question, how does septin 7 phosphorylation by tau K2 impact neuridogenesis, experiments were performed to evaluate how neurite formation and development were impacted in septin 7 depleted cells, tau K2 depleted cells, and phosphonull tau K2 cells transfected with K67A mutant. Neurons were first dissociated from rat hippocampi, then transfected with GFP control, septin 7 shRNA, tau K2 shRNA, or tau K2 K67A mutant. Then they were replated, fixed in DIV0, 1, or 3 using fixation buffer and immunostain for MAP2, actin, microtubule, and septin 7 localization. The immunostain cells were imaged using a wide-field immunofluorescence microscope and then analyzed for cell body area, cell body perimeter, and neurite number. In subfigure A of figure 3 are the images of a DIV3 control cell and septin 7 shRNA transfected cell. Long protruding neurites can be observed when a neuron enters this stage. Per the experimental results and as displayed in both bar charts in subfigure B, average cell body area and average neurite number are significantly higher in cells transfected with septin 7 shRNA compared to control cells. By the same token, subfigure C shows an observed significant increase in average cell body area as well as average cell neurite number when cells were transfected with tau K2 shRNA and when they were transfected with tau K2 K67A mutant. In subfigure D, the percent neurite origin of each transfected cell is evaluated against the control. Cells transfected with tau K2 K67A mutant on average had more neurites protruding directly from the cell body compared to septin 7 shRNA transfected cells and tau K2 shRNA transfected cells. Figure 4 compares septin 7 localization in cells transfected with tau K2 K67A mutant and those transfected with tau K2 shRNA to septin 7 localization in control cells. In control cells, as can be observed in the image of the control cell in figure 4, septin 7 localizes at membrane curvatures and within neurites for proper outgrowth and development. However, in tau K2 depleted cells as well as tau K2 K67A mutant transfected cells, septin 7 is concentrated in the cell body and not localized as much or at all at cell membrane curvatures. We also observed that septin 7 is punctated, that is poorly localized within the neurites and lamella of these cells, possibly explaining defects in cell body outline and poorly regulated branching of neurites. Based on my experimental findings, I made the following conclusions. Tau K2 and septin 7 depleted cells, as well as cells expressing the phosphonull tau K2, develop deformities in cell body shape and morphology, as well as defects in neurite branching. In these cells, a mislocalization of septin 7 was also observed. In addition, septin 7 phosphorylation by tau K2 is necessary for proper localization of septin 7 at the cell body and within neurites for proper neuridogenesis. I would like to thank Megan Radler, my secondary mentor Joshua Oclete, and my faculty mentor Dr. Spiliotis for their guidance and support over the course of my research work. Thank you all so much for listening to my presentation. I hope you learned a thing or two from it.